Bismillahirrahmanirrahim. The reactions of the urea cycle are aimed at the disposal of ammonia in the form of urea. Ammonia is highly toxic as it can impair the functioning of the central nervous system. Ammonia is mainly derived from catabolism of amino acids and is transported in the blood in the form of glutamine and alanine. Let's have a bird's eye view of the pathway of the urea cycle. At organ system level, it is occurring in the liver. At cellular level, first two reactions occur in mitochondria and rest of the reactions occur in cytoplasm. Its purpose is conversion of the highly toxic lipid-soluble ammonia, which can cross the membranes easily, into a harmless and non-toxic water-soluble product urea, which is excreted in the urine. A total 3 molecules of ATP are required in this pathway. It is regulated by 3 mechanisms: feed-forward regulation, when availability of the substrates drives the reaction in the forward direction; allosteric regulation by N-acetylglutamate; and regulation by the induction or repression of urea cycle enzymes. Its disorders may be congenital due to enzyme deficiencies or acquired due to hepatocellular damage. The two nitrogens in the urea are derived from ammonia and in the first step of the urea cycle, carbamoyl phosphate synthetase 1 or CPS1 catalyzes the formation of carbamoyl phosphate from ammonium and bicarbonate ions. During this reaction, the cleavage of the two molecules of ATP is required to form the high energy bond in the carbamoyl phosphate. In the second step of the urea cycle, ornithine transcarbamylase catalyzes the formation of citrulline. For this reaction, ornithine is required. Ornithine is regenerated during each turn of the urea cycle in the cytoplasm. This ornithine is transported into the mitochondria through a transporter in the mitochondrial membrane. Once in the mitochondria, it reacts with the carbamoyl phosphate. The energy required for this reaction is provided by the high energy phosphate bond in the carbamoyl phosphate. It reacts with carbamoyl phosphate in the presence of ornithine transcarbamylase to form citrulline. Citrulline is transported out of the mitochondria into the cytosol through the same transporter which was used by the ornithine. It is transported out of the mitochondria in exchange for ornithine. So, here we can see that it enters into the cytoplasm. In the cytosol, citrulline reacts with aspartate, the second source of nitrogen for the urea synthesis, to produce argininosuccinate. This reaction is catalyzed by argininosuccinate synthetase and is driven by the hydrolysis of ATP to AMP. Argininosuccinate is cleaved by argininosuccinate lyase to form fumarate and arginine. Fumarate is formed from the carbons of argininosuccinate provided by the aspartate. Fumarate is converted to malate, which may either be used for gluconeogenesis or for the regeneration of oxaloacetate. Arginine, which contains the nitrogen derived from the ammonia and aspartate, is cleaved by arginase, producing urea and regenerating ornithine. Urea is produced from the guanidinium group of the side chain of the arginine. The portion of the arginine originally derived from the ornithine is reconverted to ornithine. Ornithine is transported into the mitochondria in exchange for citrulline. This completes the urea cycle. Now a quick recap of the pathway. Ammonium and bicarbonate ions form the carbamoyl phosphate. Enzyme is CPS1. Carbonyl phosphate reacts with ornithine to form squalene, which enters the cytosol and reacts with aspartate to form argininosuccinate. Argininosuccinate is cleaved by argininosuccinate lyase into fumarate and arginine. Arginine is cleaved by arginase to release urea and regenerate ornithine.